some more information about it here. This is a decision support tool to help land managers with seed provenancing and planting choices, including assisted gene flow as well as other types of choices. This is a new web app. The public version was released a few months ago, and we're still kind of collecting initial feedback and making some final changes and working on the manuscripts. This is a really perfect timing, both for me to share a little bit about with you, but also to get feedback and additional ideas for how we may be able to refine the tool to make it more useful for people who are out there actually practicing some of these ideas. This project's a collaboration. It's co-developed between some folks at UC Berkeley, where I did my PhD and went for the postdoc, and folks at GVNRA, National Park Service. So thanks to all these collaborators here who contributed in different ways, as well as funding sources. Pictures here from the nursery greenhouse operations in the Presidio, where they grow out native plants for restoration projects here in the Bay Area. So a quick overview of the talk. There's three different sort of features that I want to talk about that are relevant to assisted gene flow and climate change adaptation management decisions in different ways. I'll talk about each of these. It can be used to identify environmental zones to help, different choices around seed banking and provenancing. There's another function that helps to estimate climate change exposure to try and identify which populations across the species range may be most vulnerable and most in need of different interventions, including assisted gene flow. And finally, you can use the tool to match historic and future environments across populations to guide assisted gene flow efforts. Before I get into the details, just a little more background here. So this is an interactive web app. It processes environmental data based on user settings and returns maps and other sorts of data visualizations that users can download and interact with. What makes the tool unique is a few things. There are a number of other tools that sort of nominally do similar types of analyses. This tool, we focused on climate variables that are particularly important for California plants, including climatic water deficit and AET and some other important climate variables. We also have a lot of soil data underlying this tool, which I think is relatively unique. We know that in addition to things like climate, soil patterns can be super important for local adaptation and other plant ecological processes. So it's exciting to be able to include that in this tool as well. And then finally, it's also built on top of species distribution models for more than 5,000 native California plant species. We're not using the distribution models to try and predict climate change vulnerability. They are useful, though, as estimates of the current range of most vascular plant species across the state, which helps to sort of narrow the search space for looking at different populations across the state. It helps to give some indication of the realized climatic niche breadth or soil niche breadth of different species, which can help to contextualize some climate change metrics. And these distribution models can also help us to do some simple sort of modeling of possible homogenizing gene flow at different scales across the species range. And that's the other piece that kind of makes this tool unique relative to some of the other tools out there, is how we handle this question of the spatial scale of adaptation. Right, local adaptation means that populations often perform best in the local environment where they originated from. But that begs the question of what we mean by local environment, right? To actually quantify the environment, we have to define what we mean by the local population. And it could be anything from the hyper-local microsite where those particular seeds were collected, if it's a species where there's really fine-scale local adaptation, all the way up to hundreds or even thousands of kilometers could be the relevant scale of climate data to think about what the optimum for a species might be, or a population might be, if it's a species that has a lot of large-scale gene flow that's homogenizing adaptation patterns over large spatial scales. So it's kind of about the strength of selection, which is driving local adaptation, versus gene flow, which may be homogenizing and swapping the local adaptation. And we know that that's going to depend on a lot of ecological characteristics of the species related to dispersal and pollination modes, reproductive systems, annual, the generation time, etc. So ranging from, maybe some annual gravity dispersed, insect pollinated, herbaceous species on one end of the spectrum that could have the potential to have super highly localized adaptation scales, all the way up to maybe some things like wind pollinated trees, which you're maybe talking about tens to hundreds or thousands of kilometers of high frequency gene flow that could be washing out super local patterns. So the way that we deal with this in seeds of change is through this parameter that we call the smoothing radius, and we allow the user to adjust the amount of spatial smoothing in the underlying environmental data for the historic time period that's used to estimate the climate or soil that a given population is adapted to. And by adjusting this, they can, you can account for the biology of your own species, as well as kind of do some sensitivity analyses to think about how assumptions about the spatial scale of gene flow might influence your results. Here we're looking at coast live oak, Quercus agrifolia, a slice of its distribution here in California. And these are two different spatial scales of gene flow. The color here sort of represents the climate that the population in each spot is adapted to. You can see that changing the scale can really change your assumptions and results about what the environmental value you should be using for a given site is. Okay, so now returning to these three key functions that help with different decision-making processes around climate adaptation. The first of these is seed zones. So seed zones can help to identify environmentally distinct regions within a species range. We use clustering analysis in environmental space to maximize the between zone variation and minimize the within zone variation, identify zones that have relatively homogeneous soil and climate properties within a seed zone. Here again looking at coast live oak in the Bay Area. You can look at maps of the seed zones as well as kind of visualize them in environmental space. And these can be useful for identifying regions within which it's perhaps safe, relatively speaking, to move genetic material without disrupting local adaptation patterns. Seed zones are also useful for things like seed banking efforts or admixture provenancing efforts that actually aim to collect diverse seed from different types of zones across the species range. So the users can set the number of zones, set the balance between climate versus soil, and can also set the smoothing parameter and get different results out of this tool for any species of California plant. So here's an example again of how the smoothing parameter, those assumptions about the spatial scale of gene flow versus selection can affect the results of the tool. So on the right, we have a very broad scale sharing of environmental information across sites, whereas hyperlocal adaptation on the left, and you can see very different patterns in terms of how fragmented and interdigitated the climate zones are. Okay, so that's seed zones. The second type of analysis that this tool supports is we help identify populations that are perhaps most threatened under climate change by using something that we call a climate exposure index. So we calculate a multivariable climate exposure metric that integrates projected climate change across all 5 of the climate variables in our tool. The user can, for any of these analyses, select particular future climate scenarios and GCMs and future dates and things like that. So it's a multivariate climate change signal, and it's standardized by the niche breadth of the species, which helps to sort of put that in context of relative to the variation across the species range, right? If you know something about the level of local adaptation in your species. Maybe you have some hypothesis that the niche breadth of individual populations is 50% of the niche breadth of the species as a whole. And so this type of like standardization by species realized niche breadth can help put some of the projected climate exposure in context. And if we look here for coast live oak in the Bay Area, we see here a pretty strong east-west gradient in the predicted climate exposure. Areas along the coast, buffered by the Pacific Ocean, predicted to have much less extreme climate exposure compared to inland areas. So we have coast live oak has a few populations in the Sierra foothills, otherwise restricted to the coast ranges, and those are expected to have like 3 times as much climate exposure integrated across the 5 climate variables by the end of the century. And so, you know, those foothills populations in the upper right corner, those might be places to focus management interventions for assisted gene flow or other interventions to help with climate change adaptation. And then finally, we have transfer matching. This is in some ways the core function of the tool with respect to assisted gene flow at least. Helps to match seed collection and planting sites, accounting for climate change. So for a selected site, the tool calculates environmental similarity to all the other locations across the species range based on climate and soil data and spanning 2 points in time with respect to climate. So for this function, you select a particular focal population, and you can either model sort of the climatic similarity or soil similarity of inbound or outbound gene flow to your population. So here in this example, I've selected this foothills population of coast live oak we were talking about. Setting this as the planting site will model the suitability of inbound gene flow from other spots across the species range. Look at how similar the historic smooth climates of other populations are to the future climate of the planting site. And what we see here is there's basically nowhere that has that really low blue value that we would like to see as far as like places that could be good sources of inbound gene flow into the foothills populations. If we look at this in environmental space, we can see why. So this plot on the right shows a couple different climate variables. The point cloud is the historic climate of all the populations of the species across California. And a little red quiver of arrows shows the historic and predicted future climates of the selected site. And we can see that this site was already on the very edge of the climate envelope of the species and is expected to get even more extreme in that direction. So there's probably not anywhere that's going to have a historic climate that's similar to the future climate within the species range. So that's unfortunate, but still useful information. But if we invert things and set this as a seed collection site, and then look at the suitability potentially of moving genotypes out of this foothills population into other parts of the species range, we do see a lot of really close matches. So, you know, all the sites in blue here have future climates that are similar to the historic climate of this foothills site, as well as being similar in terms of their soil characteristics. So those are potentially good places to think about as candidates for assisted gene flow from our isolated foothill populations into those sites. And that could be a way to potentially, you know, preserve some of the unique genetic diversity that could be present in that foothill site if it is possible that might go extinct under climate change, as well as you know, helping maybe bolster the fitness of some of the other sites found in blue. Of course, you know, there's all the caveats and risks associated with assisted gene flow. So thinking about, you know, outbreak depression, thinking about the possibility of interfering with other sorts of local adaptation things is important. But this can hopefully help be a piece of information that managers can use as they work through some of those decisions and trade-offs. So that's Seeds of Change. Again, hoping these sorts of analyses can help to inform some decisions, but also open feedback about ways we could improve or modify these to make them even more useful. So the tool's online at this URL. We're working on a shorter URL and a faster server, so